Okay. Yeah, one more minute. All right, let's get started. It feels like it's been a long time since I was here. All right, one today and then Wednesday, and we'll be done. Semester is over. Okay, so immunoassays is the topic of today's lecture. So just a little bit of background information on concept and theory. So this is, immunoassays are based on interactions of antigens and antibodies. Antibodies basically are protein made up of protein polymers. So here what you see here, 4 polypeptides, so the heavy chain and the light chain. And here is the binding site. And usually the binding site is as large as about 6 glucose units. And here you have the antigen, and the epitope is where the antigen binds to the binding site. And you have 2 binding sites per 1 antibody. So antibodies are produced by B cells. So when an antigen or a foreign body enters our body, B cells can produce antibodies to them, fight them as foreign matter. And the next time we ingest that protein or foreign matter, we might develop an allergic reaction because now we have antibodies to this foreign body in our system, and an allergic reaction occurs. So basically, it's an interaction between a foreign object and an antibody to this foreign object. But in assays, it's not a foreign object, so we have antigens. They could be large molecular weight antigens such as protein, or they can be small molecular weight antigens such as mycotoxin, for example. So we can use antibodies and immunoassays to detect large proteins or small molecules such as pesticides and mycotoxins, for example. So the antibodies interact with the antigen via very strong interactions, but they are non-covalent interactions. They are very strong non-covalent interactions, very unique, and among the strongest non-covalent interactions between known molecules. The affinity constant is so high, which is the inverse of dissociation factor. So it's really, really high, the binding affinity. And the interactions are non-covalent, so they're ionic, hydrogen bonding, hydrophobic, and van der Waals. So all types of non-covalent bonding. So we have the binding sites, and every antibody has 2 binding sites, and then you have the antigen with epitopes, or the region where the binding happens. It can be a linear epitope. Let's say if this is a protein, a linear epitope, let's say this is amino acid 1, 2, 3, 4, 5, they are in sequence, so linear, and can bind to the antibody, or they can be conformational. So 3 amino acids here and 2 amino acids here form the epitope, or the region where it binds to the antibody. So here's an illustration of a linear epitope on a protein and a conformational epitope on another protein. So that's what it means. And then another important terminology is that we have polyclonal and monoclonal antibodies. So what are these? So polyclonal antibody, let's say a foreign protein or a protein that we are going to be allergic to, like this is a protein molecule. And then you have an epitope here, an epitope there, there. So our bodies produce antibodies that can bind at different locations on this protein. So these are polyclonal antibodies. They bind at different epitopes on that one antigen. Monoclonal, you have your protein, and then you have a monoclonal. That means all of the antibodies bind to the same location. They can only bind to this one location. So this is monoclonal. Now, polyclonal can be formed in a host. So let's say the goat, we inject the goat with the protein, the goat generates antibody. We can take a sera, and the sera will contain polyclonal antibodies. The monoclonal antibodies are formed by B cells that are cultured. So B cells are harvested from an animal source, and then cancerous cells are fused with B cells so that they can multiply and grow. And different B cells will give you different antibodies. So a single type of B cells will generate a monoclonal antibody. So we can use those in assays as standard reagents. So these are important to have monoclonal as standard reagents. When you get an Eliza kit, you most likely will have a standard reagent of monoclonal antibodies. Polyclonal, we get them directly from the blood of the host. For a human, we want to get antibodies from the human. We did that in a study for soy protein allergy. So we took the soy protein, we modified it, and we got sera from individuals that are allergic to soy protein and determined that the modification we have done impacts the immune activity, reduces the immune activity. So we worked with polyclonal antibodies. So there are specific consideration when we're using immunoassays. So first of all, immunoassays are specific. So antibodies to the antigen, very, very specific reaction. It's very sensitive, so we have low threshold and very simple with a lot of kits out there that will potentially can be used for our assays. So we need for the immunoassay to work, so we need the correct antibody, the proper antibody for the antigen of interest. So if we're looking at an allergen, we need specific antibody for that allergen, whether it's soy, peanut, egg, whatever it is. So we need to correct antibody for the antigen of interest. We need to be able to separate and differentiate from bound antigen. So we need an assay where we can separate the bound antigen, that means antigen bound to the antibody, from the free antigen. So I'll show you what that means in a second. And we leverage a very characteristic feature of a protein that is the ability to bind hydrophobic surfaces. So that's what meant bound antigen or bound antibody that bound to a hydrophobic surface in a plate, for example, where we run the assay. So the plate, the material is hydrophobic. The protein or antibody can bind and get fixed on that surface, which is very important for the assay. So how do we produce antibodies specific for measuring antigen? I said that already. We inject it to a host, either a rabbit or a goat or a sheep, whatever it is, or even a human. We don't have to inject the antigen. If we are allergic to certain proteins, we can get the sera and we will have antibodies specific to the target antigen. Okay. What are some applications for immunoassays? Where would they be beneficial in food analysis? Where do we use immunoassays? For what, Paige?
Because, yeah, allergen testing.
Allergen testing, that's one. What else? Other than looking at presence of allergens, what else can we use immunoassays for? GMOs, presence of GMO, you have unique genetically modified proteins that can indicate that this crop or whatever it is genetically modified. What else? I gave another example a few minutes ago. What can you use immunoassays for?
Was it like pesticides and mycotoxins?
Yes, pesticides, toxins, basically mycotoxins. What else? Anything you can think of? Finding horse meat in beef patty. So horse protein. So you can detect the presence of foreign meat in what's supposed to be meat from beef or pork. Detection of bacteria and viruses. So lots of different applications. Allergens, meat species authentication, GMOs, toxin identification of bacteria and viruses. So these are very common assays used. So immunoassays started by using radioisotopes. So they would use the iodine isotope and link it to an antibody, and then the antibody binds to the antigen, and then they measure the isotope by radiation, radioactive detection. But because of radioactivity, they wanted something that is easier to work with and not having to have necessary laboratory for it. Then they figured out binding enzyme to the antibody or binding enzyme to the antigen. So now they became enzyme-linked immunosorbent assay, ELISA. So enzyme-linked immunosorbent assay, we have different kinds. We have the sandwich ELISA, indirect ELISA. So sandwich ELISA can actually be direct and indirect, and we'll talk about that. Competitive ELISA, mostly for small molecules. And then other immunobased analytical assays, purification using affinity chromatography, lateral flow strips that you can measure, detect GMOs on the field. If you don't have laboratories, it's a good qualitative method. Or Western blot, which is also a good qualitative method, but it's much more involved and involved assay compared to lateral flow strip. So using enzymes for detection, so they link the antibody or the antigen, we link it with an enzyme, covalently link it to an enzyme. And usually we use most common enzymes is horseradish peroxidase or alkaline phosphatase. They are very reactive enzymes. They can convert the substrate to a product in a reasonably short amount of time and gives you a product that is colored. So we measure by spectroscopy, basically. And using a plate reader. So your spectrophotometer, instead of having a cuvette, you can put a whole plate, 96 wells, and you have so many different samples in that plate, and you can get instantaneous measurement of so many samples in one plate, and that's that. So you would have your reaction occurring in each of these wells. So you will have your standard, your samples, your replication, all in one plate. And you will put that plate in a microplate leader, and it's basically a spectrometer, but instead of a cuvette, you have a plate, and then each well, the absorbance is measured in every well, and then you get a sheet with all of the absorption in each well at one time. Very convenient, very rapid, especially when you have so many samples to run. So two types of ELISA. You have the direct. So here's your antigen, here's your antibody, and here's your enzyme or label. Usually it's your enzyme. So, and then you add a substrate, the enzyme convert the substrate to product, and then you measure the absorption. So here, basically is your antigen immobilized on a hydrophobic surface, and then the antibody reacts with it. And if the antigen is present, the antibody remains after you do some washes, and then you get a color generated after the substrate is added. Indirect ELISA is very commonly used because it's easy to purchase antibodies already labeled. Instead of having an extra step to label the antibody in the lab, which takes a little bit of time, you can purchase an antibody that is labeled already. So what you're seeing in this image is you have your antigen, the antibody to the antigen, and then antibody to the antibody. So what does that mean? So let's say this is a protein that is, say, soy protein. This is an antibody we got from people that are allergic to soy protein. So in their serum, in their blood, they have antibodies, and we use the serum to react the antibody again. This is not labeled. We can label it ourselves, but it's an additional step. So we get antibody to human serum. So how do we get an antibody to a human antibody? You inject a rabbit or a goat with human antibodies. Then they generate antibodies for that antibody. We collect the sera from the rabbit or the goat or whatever animal host, and we add the enzyme to it, we conjugate the enzyme, and then this becomes a reagent that we can buy. So we can buy antibody for human antibody. We can get the antibody, human antibody from serum for our study, from blood, from donors. And then we can perform the assay much easier than us getting the human serum, for example, and linking the enzyme. So oftentimes our assays are done via indirect detection, antibody to the antibody. So main steps. So I'm going to draw what happens as you build your ELISA. So you have your surface, which is your microplate that you are putting your sample in. So that's hydrophobic surface. And if I'm going to do sandwich ELISA, I will start putting my sera or antibodies to the antigen of interest. So I will add to the hydrophobic surface my antibodies. So they will bind their proteins. So they're going to bind via hydrophobic interactions to my hydrophobic surface. And then I have a wash step. So I put a wash solution that washed everything that did not get bound up. So I now have antibodies bound here. Now I have a lot of hydrophobic surface still. If I add my antigen, let's say my antigen is protein, if I add my antigen, the antigen might bind non-specifically to the hydrophobic surface. I don't want that to happen. I want my antigen to bind specifically to the antibody. So what I do is I add a blocking reagent, block. My blocking reagent oftentimes is bovine serum albumin or gelatin, any type of protein or antigen, any type of protein that is not my antigen. So it will bind in excess. So I added in excess. So it binds all of the hydrophobic surface. So now I blocked it. So the antigen can only bind specifically to my antibody. Every time I add a reagent, I have to have a wash step. So I add BSA, bovine serum albumin. I incubate for a little bit. They bind non-specifically. They won't bind to my antibodies because antibodies are specific to the antigen. They're going to bind to the hydrophobic surface, blocking that. I do a wash. Everything that doesn't bind comes off. Okay, so now I add my antigen. My antigen is going to bind here. Anything else I have in my sample will be washed off. So now my antigen bound, anything else is going to be washed off. Then I add another antibody solution. So this forms my sandwich Eliza. So my antigen is sandwiched in there. This is labeled though. It can be labeled. That would be direct sandwich Eliza. I add my substrate, and then it gives me my product that is colored, and then I can measure the color absorption. At every step, there's a wash step. So anything not bound will be washed. So every time, so I add a reagent, I let it incubate, then I wash. Then at the end, I have my detector or my enzyme linked antibody, and then I get the substrate converting. I add the substrate, obviously, wash, and then the substrate will react with the enzyme to give me a color to measure absorption. So that's what the slide is telling me. And so this is an illustration from, it's in your book. I just added the images here. So this is your sandwich Eliza. One thing that we can purchase a labeled antibody that can have a magnified signal. So the antibodies can be biotin-related. So you have biotin molecules attached to it, the vitamin biotin. What we can then do, we will have streptavidin, avidin, that is. Avidin is a protein, streptavidin will be bound to my enzyme. So that then that can bind biotin. Avidin binds biotin. So the avidin binds to biotin, and then I will have multiple enzymes at one antibody. So I magnify the signal quality. Having a lot of enzymes at each antibody, I can enhance the sensitivity of my assay. So this is an illustration of what I drew earlier. So you have your solid phase or hydrophobic surface. You have your captured antibody. So if in an exam I ask you to label, this first one is the captured antibody, and the one that is labeled with an enzyme is the detection antibody, because it's linked to the enzyme and the detection happens here because of the conversion of substrate to product. Okay, so solid phase or hydrophobic surface, capture antibody, my sample containing everything, including my antigen, the red will bind. I do a wash step, everything gets washed off. I add my detection antibody that has the enzyme. It binds, it sandwiches. So mind you, it will bind at a different epitope. You have an epitope for this specific antibody, and then there is an epitope here for this detection antibody. And then you get the conversion and a color is formed. Here, in this case, the higher the absorption, the higher is the amount of antigen I have in the sample. The more antigen I have, the more detection antibody is found, the higher is the color, more conversion of product to substrate. So the higher the concentration of the antigen, the higher the absorption. Yes.
I make sure I have this straight. So the first antibody that we captured, that's what we're just trying to figure out if it's there naturally, or we added that antigen and we added the detection antibody.
No, the antigen is what we're after.
Oh,
We are detecting the presence of this antigen, whether it is an allergen or another protein, a horse protein or pork protein. We are detecting, this is what we're after. We're after this. So this is just part of the assay and capture antibody that is specific to my antigen. So I need to know what my antigen is, and I need to get antibody specific for it.
it's not captured, it's there to be captured. The antigen. When it's captured.
Oh, it is capturing, yes. So
Okay,
the
okay, yeah.
captured antibody is the antibody that is capturing the antigen.
Okay. Thank
Yeah.
you.
And it is linked via hydrophobic interactions with the solid surface. Those antibodies are proteins as well. Good. Now the indirect Eliza is what I explained. We get an antibody to the antibody. That means if I have a human antibody and I'm using it to sandwich my target protein, then I get a human antibody or antibody to the human antibody. Antibody to the antibody, we call it sometimes secondary antibody. Okay, when we say secondary antibody, that means it is an antibody. Then this human antibody in this example is the antigen to this. Am I confusing all of you or not? Monday. Okay, so again, I'm going to repeat. Let's say this is soy protein again. Oh, we have the quizzes. Yeah, you already saw your grade already. Okay, so Brigida, you can distribute. Okay, so let's say this is soy protein. I got human serum, allergic to soy from human donors. They donated their serum. So we first put the human serum from that has the antibody on the plate. We let it bind. Then we add our sample, our modified protein, let's say. We add it, it binds. Then we add another layer of human serum. Mind you, we use the blocking PSA. Always remember you to block so that you avoid non-specific binding. And then the human serum again has the antibody that reacts at a different epitope region on the protein. And after that, we get a labeled antibody, or we call it secondary antibody. The secondary antibody is basically an antibody to the human antibody. So let's say we injected a goat with a human antibody, they generated an antibody. So this is a protein. You always have to remember antibodies are protein. So they can be antigen themselves. So this we can purchase already labeled. So the secondary antibody already labeled with the enzyme. Then we add a substrate that converts to product. The same thing, with higher concentration of target protein in my sample, I get higher absorption. Both direct and indirect, higher absorption means higher concentration. The difference when we say direct and indirect, the indirect is the labeled antibody is an antibody to the sandwich mixture. So here you have the antibody to it that is labeled. Yeah.
And for the structural component, so is it the detection antibody that's like detected?
No, what's detected is the color because the enzyme here is once everything is washed off, if we have a protein, this is going to remain on the surface of my plate. If I add a substrate, the enzyme is going to convert it to a product. I have a change in color, and then I measure it by absorption. If I don't have an antigen, everything will be washed off. When I add a substrate, there's no enzyme, so there will be no color.
Okay, that makes sense. Thank you.
Yeah. So comparative ELISA is specifically for small molecules. So pesticides, toxins, anything small. So when you have a small molecule, you don't have a big protein, you don't have multiple epitopes. So when you have a small molecule, the entire molecule is bound to your antibody. So there is no other epitopes. So you cannot have a sandwich ELISA because there is not multiple epitopes where one antibody is bound at one region and another one sandwiches it. We cannot. So we cannot use sandwich ELISA for small molecule. So if in the exam I give you a sandwich ELISA figure and I tell you to label it, and then I tell you, give me an application, and you say mycotoxin, it would be wrong. It cannot be used, comparative ELISA cannot be used for large proteins, and regular ELISA cannot be used for small molecules. So sandwich ELISA cannot be used for small molecules. So since it's a small molecule, and if we want to generate antibodies for this small molecule in a host animal, we have to have a carrier protein. So it's really small, it's not recognized as an antigen. So in order for it to be recognized as a toxin, as an antigen, it has to be carried by a carrier protein. So the carrier protein, so I would have a protein here, and we paint this to a triangle just for ease. I will have a carrier protein where my toxin or small molecular weight antigen is bound to it. Then this gets injected in the host. So then the host detected as a foreign matter, then I will get antibodies to the whole hapten. Hapten is this. It's the antigen of interest and the carrier protein. So the reason I have carrier protein, because if I inject only the small antigen into the host, it won't recognize it. it has to be carried with a bigger molecule. So we use proteins, albumin proteins specifically, to generate antibodies for different locations, including the actual small molecule of interest. So what happens here in ELISA, the competitive ELISA, I have a hydrophobic surface. I have antibodies specific to my antigen, and I have the hapten that is labeled with an enzyme. So what happens here is that I have the hapten labeled with the green enzyme. I have my sample. I put them together, they compete. That's why it's called competitive. They compete, the antigen that is labeled with the hapten and the enzyme, and the antigen read from my sample. So they compete for location on the antibody. And then when you wash, you add the substrate, and then you generate coloring. In this case, the more antigen I have in my sample, do I have higher absorption or lower absorption? With more antigen in the sample, do I have higher or lower absorption? Lower, yes, because it's competing with the labeled one. So the same antigen I have in my sample, I have it linked to in a hapten form, linked to the enzyme. So the more sample antigen I have, the more of the antigen in the sample will be bound, and the less of the enzyme linked one will be bound. Therefore, I would have less absorption. So here, it's an inverse reaction between, it's an inverse relationship between concentration and absorption. So we can have different formats. We can have a bound antibody format for a competitive ELISA, or I can have a hapten bound format. So let's start with this one. If I have the antibody that I bind first to my surface, then I have my labeled hapten, and I have my antigen in the sample. Again, there is a competition happening. The more antigen I have in my sample, the less labeled will be bound. And the less antigen I have in my sample, the more labeled hapten will be bound. So here, my label is antibody. The enzyme is attached to the antibody. And I have a hapten, which has the carrier protein and the antigen of interest. And then I have my antigen in my sample, the free antigen coming from my sample. So in this case, the antibodies that capture antigens from the sample will be washed off. The antibodies that bind to the hapten will remain, and then the enzyme converts substrate to products. So again, here there's competition between the free antigen in my sample and the bound antigen on the plate. The more I have free antigen, the less I have color. The less I have free antigen in my sample, the more antibodies that are labeled will bind and remain and give me color. One thing here to pay attention, the protein used to bind the antigen in the assay must be different than the protein used in the animals to produce the antibodies. Because you don't want the antibodies to bind to the protein in the hapten. So we want to be sure that whatever protein I use to generate the antibodies in the host, these carrier proteins should be different so that the antibody can only bind to the antigen, not to the carrier protein. So that is a must, otherwise the assay will not work. Here's an example of measuring presence of aflatoxin. So if I have a lot of aflatoxin, high toxin here, I have low absorption because I have less labeled bonds because it's competition. When I have the highest absorption possible, that means I don't have any aflatoxin in my sample. All of the bound or the enzyme-linked captains are bound to the antibody. Therefore, I have really high absorbance. So again, in competitive ELISA, higher absorption means lower concentration in your sample. Questions? Okay. Immunoaffinity purifications, like your chromatography, here you have your silica, and with the silica you have antibodies are bound, and you have your sample that has your antigen of interest and other components. Your antigen will bind specifically to your antibodies that are immobilized in your column. The other non-specific protein will be used, and then later on you can elute your antigen by changing the ionic strength, pH, or eluting solution by having antibodies in the buffer. Therefore, they can leave the column and you can collect. Okay, chromatography again. This is a lateral flow strip. This is usually used on the field if we want to determine presence of GMO. So what happens, this is kind of similar to the pregnancy test. So what happens here, you have your antigen in your sample, in your liquid, and you dip your strip in. If you have the antigen, they will bind, and your antibody has a color attached to it. Then they will move up the strip by lateral flow, and they reach to a place where you have another set of antibodies bound, and then you get sandwich ELISA. So the antigen is sandwiched here, and you see a color. So in the actual strip, you always have a control line to see if the liquid has moved and there are no blockage. Determine that it's working. So again, you dip your strip in your liquid, and if you have the antigen is going to be bound. Your antibodies bound with the antigen, flow up by lateral flow. They get to a place where you have the antibodies that are immobilized. And then if you have the antigen, they will be immobilized here, and you see the color. The control line, you have here antibody to the antibody. So these color labeled antibodies, in the control line, you have antibodies for them. So as they move up, they get captured by antibody to them, and then you see a color. So you have a positive test when you get two colored lines, and you have a negative test when you only have color at the control line. So the control line tells you that the strip is not blocked, it's working fine, and you have no positive antigen, so you don't have GMO in this case, in this example. So negative versus. Okay, it is a sandwich ELISA again. Now, immunoblot or Western blot specifically. So what you do here is you run a gel, and usually a 2D gel, because then you can separate the proteins even more, and you have spots of your proteins separated, and then you transfer them to PVDF membrane. So you use a sponge, and you put your gel, you put your PVDF membrane, and then you sandwich them, and then you run a current, and the proteins transfer from the gel to the PVDF membrane. Then you add primary antibody. If the antigen is there, it will react with it. Then you add your secondary antibody that is labeled with the enzyme. So it binds to your primary antibody. You add a substrate, it gives you color. Then you can see if you have the antigen, you'll see that red spot or whatever color the spot is. You'll see it on the gel. So this is here an example of it. So here we're running pork, different types of pork, beef, horse, deer, chicken, turkey, whatever, duck. And you want to determine the pork protein, so you have an antigen for it. Then when you do the transfer and then you incubate with the antibody for the pork, then you only see a color attenuated and that band, specific band, and nothing else reacts. That's one way of authenticating the source of protein you have in your meat sample. Questions? We ended 5 minutes early, but okay. So what we're going to do on Wednesday, we'll have a review session, but before I start the review session, I want to run through one lecture because I'm behind 20 minutes in lecture. So I'll go over lecture material, then we'll do review, and then we'll have our last lecture in class here on Wednesday. Please review the questions that I put up on Canvas, I believe under week 15. Please review them, try to solve them. Come with questions. I can answer anything you have, any concept. We can go over anything before the final. For the final, it will be on everything from the beginning of the semester with the focus on the material that were not in the midterm. But obviously, sampling is important, data analysis is important, chromatography, spectroscopy. So you need to know these because they are all applied in the later section of the class. Valerie? Where? Oh, in lab. Yeah, ABLMS 135. Yes.
And is that going to be during lab period? So
During the lab period. So you need to come at 9:30, like you have a lab, we'll do a quick lecture and then we'll do review. We still have class? Yes, I'm behind about 20 minutes in material. That's why I'm going to use some of the review session for that. And then we'll have a lecture.
then the final exam is this time on Monday?
Not this time. Okay,
What time?
everybody, what time is the exam? 8 a.m. here on Monday. Yeah. They should know that there's a final, but I'll give you time. Yeah, yeah. Oh, extra credit. Did you separate them from me? Yeah, I did. Can you separate them? Oh,
I also have my lab notebook.
so what is this then?
Those are both in class. This is last period and that was earlier. You had said to hold on to that one initially, but
That's from which chapter? pH and particular acidity?
yeah.
Okay, okay. All right.
And I just have my lab notebook.
Oh, the lab notebook.
Yeah.
Nigel, where is Nigel? You have the lab notebook thing? Thank you. Lab notebook.
Oh my goodness.
10?
I can grab it.
Oh, pencil, yeah, that would be good. I don't have anything to write with. So what was the situation there? I just- Check on Friday. Okay.
This is the file, and then Lenny is not here, so.
Oh, yeah. All right. Okay. Sounds good. Thank you so much. All right. So which one do we have to look at? Protein, carbohydrates, fat, carbohydrates. 65. Thank you. All right, so from my protein, 39.
Which one's the pH? Like the it's just I just have tape, so there's
Yeah.
a little
Yeah.
yeah, it's interactive.
For sure. All right, thank you. Valerie, I'm gonna keep your
You
pencil
can keep
for
it. I never use that one anyway.
Valerie.
I'll put it there too.
The video was saying different.
Yeah.
Let's look at that. All of this here, okay. Two groups. Two groups for carbohydrate. And some people will be just standing doing nothing. Potentially. What is that? For extra credit?
Yeah.
You want to put them somewhere there? Okay. Okay. So. Where is the calculation? For the protein.
So that one.
Unless you,
I think I combined these two.
The lab
I did,
report,
they were combined labs, so I just did them all in one.
where do you have all of them?
There's some.
That's nature. That's fast. Oh, that's the percent nitrogen. So, all mixed. Okay. All right. And then carbohydrate. Calculation.
And then there's just a few page break.
That. So
And
calculation.
there's more. It's just I did a big page break to make sure I'd have enough time.
Oh, so these are, that's fine. I just was looking at 3 labs now. So, number 4. Okay, thank you.
So are we gonna need to bring this? Can we just keep it?
You can keep it home. That's fine.
Okay.
I don't think I'll have time to look at the 5th time.
Oh, okay.
That's okay. Please. Where is the calculation for this lab? Okay,
I might have forgotten to add it when I was printing out my.
okay, I'll take out half a point and then you, if you want, you can bring it on Wednesday. Okay. Are these your extra? Three. All right, so we're gonna look at proteins first. So what are your calculations for this one, for protein?
If they're not in there, I might have forgotten.
Wait, so ash, substrate, no joe.
Yeah, I might have
Forgotten
forgotten protein.
protein. Okay. What? Can't forget about protein.
That's a protein I want to problem.
Okay. So I'll just take half a point.
Okay.
I mean, I can give it back to you if you couldn't show me on Wednesday. Okay, Shri, thank you.
Thank you.
It's like this guy was just doing this. And they have really nice things.
Hi, oh, look at the line. How great is that? Okay, so we are table of contents. So we are page 44. Yeah. Protein. else. Very good. Okay, thank you.
Kind of a little bit updated
You updated?
for the last time, so I got it now.
Yeah.
Here, and then I kind of add a little bit of the notes, but I'm not sure like how much are able to extract it. Like right here, I add kind of a little bit.
More complete, okay.
That's it. That's what we do.
And then you had all of the calculations for this one.
Yeah. Oh, it's 3, number 3.
You're doing, right?
Yeah. Note number 3, I got a 9.
Okay, I have it 9.5.
Okay. Thank you so much.
So we're gonna look at protein first. Protein calculation. Got it. Yeah.
So yeah, I got my little drinks, I like set up my little announcements, I was like, no. Yeah, we're here for you, man.
Yeah, I can see.
Get that ready.
So,
I updated from
oh, from last time? Well,
So hard to check
you're
that.
feeling. Oh, okay. So fat fermentation. Okay. That's the only after that I'm dating. Okay, so what was missing? Some data. Question calculations. Good. You're giving you guys a grade.
You are not going to grade the pH and
No, I'm not doing that.
I didn't realize that.
It's complete. Yeah, I know. I don't have time to go for overall. That would have been part of the 5th time. So I'm just gonna evidence what I've done so far. Okay, thank you. What? Yeah, good. You're the longest wait, because I haven't seen it twice.
Oh, I haven't.
You gotta wait longer.
I was just missing my.
What were you missing?
I wrote it down too.
Protein, fat characterization and carb.
Yes, missing, so they're permitted.
Okay. And
Yeah, from
that's it, okay. Flip the page.
I got little
Flip the
full
page.
pages for you, all kinds of stuff.
Good. You get your points back, sorry.
Thank you.
Okay, I'm not gonna write it here. I'll just give you 10 over here.
Okay, and also I forgot to bring in my +2 from Friday.
Bring it on Wednesday. As long as I have everything by Wednesday, that'll be good.
You got it.
Thank you. All right. Abby,
Yeah.
what are we missing, Abby?
I'm not sure you gotta check it. I don't know.
Abby, where are you? What's your last name?
Daddy. DADI.
Are you Wednesday or are you,
Wednesday.
you're Wednesday?
Yeah.
Oh, you're the first. Okay, so fat,
Yeah.
missing fat and sample calculation. Okay, so I'll look at fat characterization then. This is good. The calculation. Good, you get your point back.
And I don't give an understand that bonus you said you promised.
What did I promise now?
In the class you promised you're gonna
I'll-
just say.
Everybody's gonna get it.
I don't know what I was missing.
Well, were you Miss Caitlin?
I don't know. Like
Let me see what-
calculations or something.
Fat characterization income. That was a fat characterization. So.
You can see like where they.
the calculation here. Okay, good, you get your point back. You get.
That's the one thing I was missing?
That was the only thing you were missing.
Right, okay.
Thank you. Yeah. You were missing fat characterization and protein.
Just say that.
I don't know, I can't read Nigel's. I'll look at from protein. I don't know. What
Wait, no, I don't. Okay, I thought I was,
protein?
these were all like stuck together. So I thought they were like right next to each other. I need to go. I don't know how far I need to go.
I haven't got the tables.
She's probably gonna be
Do you
mad.
have the calculation?
They're in the computers.
All right, so I'll give you a 9. If you want, you show me on Wednesday, I'll give you back this one. Okay, thank you.
All right. I think this one is a double check because I got pushed back so far with all the guest
I'm
lectures.
missing Michael. Michael gone.
Give him a 0.
And Tyler.
All right, you,
Drew.
let's do
what
the
were
check.
you missing?
I'm
Missing.
because I think you told me to check it and then I
Oh.
think you were gone. And so I missed it.
So 2 times. I
Yeah.
haven't seen since moisture, the lab before moisture and onward. I think.
You need atomic.
Yeah, the lab just before moisture.
Okay.
Yeah, I mean, just learning how to keep track of everything.
Mhm.
Data. And then moisture.
Yeah, I had the atomic absorption and I didn't have the other 2 done because I think we were working on that. So you're missing this here, other way.
Ah, that's it.
It's like a pop-up book. I tried to designate like everything with 2 pages, but then sometimes it doesn't work out that way.
Okay, so that is the first part that you're missing.
Yep.
Get you 10 for that one. And
These
the
are all
last
extra
one
credit
is
points, right?
These are not extra.
I can try. I just use the same tables.
Okay. And then the carbohydrate. Oh, you need a lot of space. Where's the calculation for this part?
Oh, I don't think I have them for this one. I think when I did the lab report, I forgot to do it. If you want to, I can get the calculations done on Wednesday if you want. Okay.
So we have calculations for this one. So 9.5. Show me calculations on Wednesday and you get yours.
Okay, sounds good. Thank you.
Yeah,
I'm not here for my lab.
I'm not here for
Sandy's
you.
on top of it. Sandy's like, I just need a
Michael.
question.
I just have some questions.
You were here for 20 minutes.
I know, well, I happened to come on the wrong day to ask a question.
Justin. Oh, Elisa? You're next, okay.
Okay, I have a few questions.
Yeah.
So my first question is like for this question on the quiz. I did not do as well as I expected. It's mine.
Oh.
So like,
for the, like, I don't know, I didn't think that was right either, but really like,
research
oh,
method,
Bradford is like, I mean, it says here it's like rapid.
it is rapid, but Bradford is not for complex food matrices. It's for usually purified samples. It's a research method.
Yeah, because I just like don't remember really learning this for protein.
It's, didn't you study the lecture, the IR lecture?
The IR one?
Yeah,
Yeah,
it was
lecture.
part of this course. Yeah.
Okay.
So, and the IR does tell you it is a fast method for quality check for all of compositional analysis, whether it's
Because really, I was
protein
just
or
like,
fat.
UMass is honestly one of the fastest ones because this one you still have to calibrate, you know, which takes a long time.
But once it's calibrated, it works for extended period of times until you have a shift.
Got it.
Yeah, yeah.
All right. Well, that was a
And
good one.
DUMAS is not usually used as quality check.
Okay, sounds
Yeah.
good.
Mhm.
Okay.
The rest of that.
And then for the lab report, like when our discussion is a page, if we add any like sources, is it okay to go?
Is it okay to go over?
Yeah, like the discussion is only supposed to be page, but when I added my in-text citations,
Yeah,
this time it knocks it off.
it's okay. I don't think Kat is gonna be mean about it.
Because I have a lot of in-text citations.
It's,
Okay, sounds good. That's all my questions.
that's it? Okay.
I know, I waited in a very long line.
That's okay, Alyssa, I think you're not hearing today because of this over your ears. Yeah. She was telling me I'm soft spoken today because she was talking to me and she couldn't hear anything I'm saying because you're covering your ears. All right, so I am missing 2 grades for you. So I'll be looking at just before the moisture. So it would be AA, page 30.
I will chose the wrong notebook for this.
It's okay. So what else? AA. Data. What is calculation? I don't see them, so half a point. And then, oh,
Oh, I do, yeah.
yeah, so you have them, no half a point. Good. Good. Okay. Where is your calculator? I don't see calculator.
Oh, I think those are just, yeah, I guess these
It's
are
okay.
numbers.
Yeah,
Yeah,
okay. So this one you get -5. And then 4 is
For the last two, I have raw data in there. This one, organized from the graphs. I don't know.
You were not there for factorization? You were not there for factorization?
But characterization No.
So we have the equations and the results. It's okay. Just when you do the lab report, just stick in your calculations when you do the lab report. Yeah, wasn't reported you to do? Yeah, did you finish it?
No, not yet.
Okay. All right, that's fine.
Yeah, I mean, I do have these calculations.
Yeah, it's okay. I'm
Okay.
I'm giving you a pen, that's fine. All right.
Yeah,
Yeah?
yeah.
What's that?
Are we counting participation for the review session?
No. I don't want to have Nigel sit. What's that? I don't want to have Nigel be tortured for 3 hours. If you participate, Saj, put it on your notebook, how many participation on rescue at the end of the review session. Yeah, I don't want to have Nigel sit for 3 hours. Yeah, I'll tell them. I don't know what I'm doing. Oh, this is so mixed up.
About changing the meeting time.
Huh?
I replied your email about changing the meeting time.
Okay.
So I thought next week would be better. Wednesday.
Okay, thank you. Oh. Oh. Yeah.
Maybe next time we can talk to the title there. Title there, information, like protein sector. This is it. This is. Oh, it's just not comfortable. This the extra credit, like from the B class, like question?
Yeah, but I didn't ask for that
Oh,
to be
okay.
given in.
Okay.
Oh, so I did not stop the recording.